Hello, and welcome once again for another episode of Profiles in American Funded Research. I'm excited to have my guest today, Fatima Foflonker. She got a PhD at Rutgers University. She did a couple of postdocs, both at national laboratories, Brookhaven and Argonne, and now is an assistant professor at Clark Atlantic University. Is that correct?
Correct. Yeah.
And I invited Fatima on today to discuss her project about gene clustering. And so I thought I would just turn the floor over to you for a moment and let you give a brief description, a brief overview of the project.
Yeah, sure. So the project I'm currently working on, that's currently funded, it's called Deriving Gene Function from Three-Dimensional Gene Clusters. I am looking at clustering in three dimensions to try to identify gene function this time. I previously, at a postdoc at Brookhaven, looked into how gene clustering can be used to identify gene function in two dimension. And that worked out quite well. So we looked at, it was about 10 different green algal species, and green algae is kind of my specialty. So we look at these green algae because they've got small genomes, and they're related to plants. And so a lot of people are interested in, kind of how we might be able to extract this information and relate it to higher plants. So we looked at 10 species of green algae in this first experiment, and then looked for gene clusters of two or more genes within a certain distance. I think it was a 6 gene distance, to see if those genes stay close together in multiple species. So, over evolutionary time, if these genes are staying close together, it's more likely that they may have functions that function together. So that's what we looked for. We did a scan over these 10 species. We found a couple hundred gene clusters. A lot of them, were completely, had completely genes completely of unknown function. But some of them we were able to kind of manually go through and get some clues as to what potential functions would be. And so, there were a couple good candidates, which there was one arsenic resistance cluster. We then were able to look at mutants in the lab and confirm that this particular gene that we found was involved in arsenic detoxification. So we were able to go from kind of like this wide scan, knowing nothing about gene functions, to filter down the possibilities, and then pick targets, and then go into the lab to confirm these gene functions. So that was quite successful in two dimensions. Just looking at gene locations on the genome sequence. And now my next step of this project, that's currently funded, is to look at gene clusters in three dimensions. So we know inside the nucleus, chromatin are folded around each other, and I'm looking for those points where they touch. We're using high-C sequencing data to try to get at those points that are close together, clustered physically.
Okay, so this
Uh,
is like a physical
mhm.
distance.
Yeah, so they should be, so the high C sequencing, what it does is um, it links together distant pieces of chromatins that are uh, basically touching or close together in the genome, and then it'll sequence just that point that's touching, so you can kind of get an idea of what genes are close together in three-dimensional space. And so using that sequencing data, I'm trying to find, okay, what points in the chromatin are touching, and is that conserved in multiple species? So we're hoping to look at 20 green algal species. So there's about 20, available in the literature right now that we're going to look at, and then also sequence some of our own. And then, hopefully we'll be able to do something similar to what I did in 2 dimensions, where we'll have a bunch of clusters and be able to kind of filter out and pick out some of the kind of low hanging fruit, to be able to select some targets for the lab or make some hypotheses on the function of genes.
Thank you very much for that introduction. It's very, very interesting topic. So, can you explain a little more about you already kind of briefly touched on it, but about why you chose green algae? Because you said that they have a small gene sequence, is that correct?
Yeah, so they tend to have smaller genomes than there's a lot of variety within green algae themselves, but some of them have some of the smallest eukaryotic genomes. So that's really interesting. These genomes are so compact. They can go from like 12, 13 megabase pairs, up to the hundreds. But some of them are so compact they're almost bacterial like. So maybe, I mean, they're still eukaryotes, right? They still have introns, but there's fewer introns, and it's just kind of giving it a snapshot of maybe kind of early evolution in eukaryotes, or highly specialized genomes that have shed unnecessary genes that might function maybe more similar to bacteria. Maybe they have more clustering. So that's one of the reasons I'm looking at these eukaryotes. Other reasons are right, they're related to plants. They'll inform us about potential gene function in higher plants. And they're also, people are interested in them as biofuel candidates. So certain green algae grow pretty fast, they photosynthesize, so, they're turning basically light energy into biomass and lipids and oils that can be extracted for biofuels.
Now you mentioned you discussed eukaryotes and you also have mentioned it a lot in like papers and things like this. Um, and can you just explain, first of all, what the difference between a eukaryote and a prokaryote is? Because we're trying to
Mhm.
hit a general audience here, but also um, what what it is that made you focus in on eukaryotes, in because you had an interesting discussion on that. I think it was in the grant description.
Yeah, so eukaryotes versus prokaryotes. So prokaryotes, that's what you think of as bacteria or archaea, and eukaryotes go, anywhere from single cellular, algae and protists, all the way up to humans, plants, animals. So on a cellular level, I would say the difference is that prokaryotes will have a lot smaller genomes. They'll be circular. Eukaryotes have longer linear genomes. And then prokaryotes tend to have, very compact genomes, and they can even, I guess the relevant part to my research is that they can have gene clusters called operons, that genes are sequentially located on the genome and they're controlled by a common promoter. So this means, so during transcription, which is when you're turning DNA into RNA, multiple genes will be transcribed into a single piece of messenger RNA. From there, basically, those multiple genes can be translated into proteins simultaneously, and it's a way to kind of regulate gene expression coordinately, right? So if you need all the genes involved in a certain pathway, they can turn on that certain pathway. I guess a very common one is the lactose operon, right? So to break down lactose, there's a couple different genes in this lactose pathway that they can coordinately turn on and off, so that they're made at the same time. They're made in similar quantities and can work together to break down lactose. So that's something that happens in bacteria or prokaryotes. In eukaryotes, they're not traditionally thought of as having gene clusters or operon, or to be very coordinately expressed like prokaryotes are. So they're, I mean, this is kind of a very like traditional point of view, but several years ago it was thought that eukaryote genes are just arranged randomly on the genome, um, and there's kind of a lot we don't know about eukaryotes. And more and more recently, there has been more evidence to show that genes may cluster, like similar genes that function similarly or in similar pathways may cluster together on the genome. They may not necessarily be transcribed into a single mRNA and translated all together like they are in prokaryotes, but they can be close together on the genome, and we may not exactly know how the gene expression is coordinated together, but we do know that they can cluster.
And you mentioned that you're going through many different um, pieces of data essentially in order to find this. So I'm assuming then that you're doing some kind of computational work. What is involved there? What kinds of techniques do you need to use in order to find these clusters?
Yeah, so yeah, I do mostly computational biology work, and so kind of the first step is to gather your like raw input data. Um, so other scientists have done, similar sequencing experiments. So basically, you figure out the sequence of the genome of interest, um, and they will post it to national databases like the NCBI, or other databases, and they're publicly available. So often my first step is to see, okay, what's already been done, what's publicly available that I don't have to redo, and download that information. And then the next step I like to do is to kind of look at a phylogenetic tree, and see, okay, what important branches or pieces of information are we missing? Like, are there any critical evolutionary branches that we don't have information for? So, I don't know if we were interested in kind of learning about the split between algae and plants, I would want, to have some information on the green algae side and some information on the early plant side. So some of there's algae on the plant side of the split. Um, so I'd want to kind of fill in that information so we can trace um, kind of the evolution of these clusters. So that would be step number two. Next, there's a lot of kind of data manipulation involved. So that would be probably data science techniques would come into play here on just basically standardizing your inputs, getting them into a form where you can write a program, which usually these days is done in Python. Um, Write a program to look for certain patterns, right? So for this experiment, I'm going to look at that IC data, so where the chromatin are touching. I'll probably need information about those genome coordinates, so where exactly they are on the genome, what species they're in, and I'll also need some evolutionary information, like what genes are homologous or similar to each other in different organisms. And so then I can basically create a program to systematically search for all those regions where homologous or similar proteins stay close together, right? And then from that, there's obviously a lot of data analysis on the back end, I guess. After you've got these clusters, okay, what do you do with them, right? So that's pretty data intensive. You start looking for gene functions that you can figure out, and usually that's from a blast search, where you would take the sequence, look at all the sequences in the database, try to match them up, and it'll give you genes with similar functions. So that's kind of one step. We also do a lot of other computational analyses, like making phylogenetic trees just out of the gene sequences to try to get a better understanding of how individual genes evolved. We look at networks to see just kind of more fine-tuned information on how these genes have evolved, and can that give us any information into gene function. We'll look at different domains within the gene. And yeah, basically it's a bunch of manual computational biology at that point, to try to really figure out what's going on.
Now, you mentioned before that for up until recently, the paradigm seemed to be that there was no clustering in eukaryotes in the genome. What are the implications if there is clustering, as you have seemingly pointed out with your 2D research at the labs, and now are looking at 3D? What are the implications of that?
Yeah, so it's pretty interesting. So I mean, and there is more and more evidence for gene clustering in eukaryotes. Specifically, fungi and plants tend to have a lot of secondary metabolite clusters. So genes that are involved in the same metabolic pathway, making some sort of compounds specific to these fungi or plants. So for me, the interesting part not only is that genes that function together are close together, and then you can infer, the function of other genes. Um, the other interesting part, I think, is that you can then think about it on from like a biotechnology perspective, that in bacteria and prokaryotes, these operons are used to coordinately express genes. So, can we use these kind of clusters, or even perhaps coordinately expressed clusters, to then, you know, synthetically, coordinately express genes, right? So we can
Can you
maybe add
can
genes.
I just
Sorry.
stop you there for 1 second? Can you explain what you mean by coordinately express?
Yeah, so we often look for ways where, so say you want to synthetically get yeast or some eukaryote to make a product, um, or increase expression of a certain product. Um, you may It's possible to kind of edit the genome, add genes, or add things that control gene expression, like promoters, so that when you kind of turn on a promoter, it's causing the expression of multiple genes at the same time. So if there are multiple genes needed to complete a metabolic pathway, all those genes can be turned on at the same time, and you can go from, your input to your output more seamlessly.
Okay, yes, thank you.
Hope that explains it.
Uh, um, and then so I know that I kind of broke you in stride there, but can you do you mind continuing on that last train of thought that you were where you were discussing about what are the implications?
Oh, right, yeah. So so I think if we are able to understand, or so first, identify and then try to understand how these gene clusters are working in eukaryotes. So part of what I'm doing is to kind of look for genetic markers associated with these genes. And so I plan to use kind of a machine learning technique to kind of input a bunch of genetic markers like promoters or where is the start codon, where is the end codon, how big are the introns in the gene, all kinds of genetic markers like that, to try to understand are there any specific features that are associated with gene clusters. Can we use those features to then identify more gene clusters in other species? So I know I kind of went off on a tangent here, but I'm gonna bring it back. So once we identify those markers, say we stumble across a promoter, so something that will turn on the set of genes. Maybe it's in common with a couple other gene clusters. We could use that promoter or those gene clusters and then synthetically edit them to hypothetically, insert genes that we want to be more coordinately expressed. So it's kind of like a more natural way of editing the genome. Um, so it could identify, certain targets like that.
I see that. Thank you. Um, what so I saw that you actually I think this was in one of the papers that you suggested. What it how well known, I thought this was an interesting detail. How well known are the proteins that are being made by these genes produced by the RNA or and whatnot? How well is that set of proteins known in the algae?
Yeah, so if we look at kind of the list of clusters that we found, some of them are very well known. Like some of them have been well researched. Like I think it's some certain nitrogen pathways that we found that were well researched. Um, some of them are known proteins, but we didn't know that they cluster together in certain species. So in one of my papers, I talk about a sunscreen compound that's well known in everything from algae to vertebrates and animals, but it looks a little different in the the specific genes. We didn't know that they cluster in some species, and they're clustering a little bit different in the algae. So yeah, some of them are well known. The vast majority of them are not. So the vast majority of them have no similar functions to other known genes. And just for context, algae in general can have anywhere from like 50 or like 25 to over 50% of their genome are just genes that have no similarity to any of the known genes identified in databases. Right, so there's a huge chunk of unknown proteins, and I guess what we're trying to get at is maybe there's some unknown proteins clustered with some known proteins, and we can try to figure out those functions.
Yeah, that really kind of fascinates me. It se- it it seems like we should know all of the proteins and whatnot, but I'm wondering if is the reason that we don't know some of these proteins or many of these proteins in algae because it's not as well studied as say like a human, or do we similarly not know a whole bunch of proteins in humans as well?
Yeah, so I would say both. I think there are um, maybe more than you'd expect of unknown genes in species that have been very well studied. Um, but also, yes, algae are kind of highly divergent evolutionary wise from its closest well-known species, right? So most of the algae we look at, in terms of algae, the best studied species is Chlamydomonas, right? And then if you kind of look further back on the evolutionary tree, you're going to kind of hit some of the well-studied plants. So if you do, like I said, one of those blast searches where you're looking at sequence similarity between different species to try to identify gene function, and you take just a random algae gene, you're probably going to hit Chlamydomonas, which is a well-studied green algae. And then you have to think, okay, well, where was its functions derived from, right? And oftentimes they're derived from higher plants, which are even more distantly related, right? So a very well-studied higher plant is Arabidopsis, and the green algae especially derive a lot of their function from Arabidopsis. Or so say and to identify gene function, traditionally what scientists need to do are to kind of knock out a gene in a species, see what function it's missing, and then they can say, okay, this gene does this function, right? So that's obviously very time consuming, very resource intensive, and there's also kind of no guarantee that if this gene did this function in a higher plant, does this gene do this exact same function in an unknown algae, right? So there's kind of a lot more unknown than you might think. And so what I try to do is kind of use those clues that you can get, like, okay, this gene is similar to a gene in a plant and it does this function in plant does, and then you can use those clues to say, okay, well, now let's verify, is that true for algae?
Yeah, and how much carryover is there between species? So if you're talking about a higher plant versus the algae, like let's say some plant that's really close, as close as you can get in some sense
Mm-hmm.
to some algae, how different are they? And then how much overlap is there in function?
Yeah, I would say there's quite a bit overlap. So a lot of the core functions, right? So core photosynthesis genes will be the same. Um, Kind of the genes that make plants, right? So there's a core set of genes that you look at that are similar in all kinds of photosynthesizing plants and green algae. So you have that core set, and then there's a lot more that are maybe algae specific or plant specific. So yeah, there is some overlap, but there's definitely a lot more that's specific to each type.
And the you're I assume though that you're not just looking at core function when you're trying to do these like the similarity in function. You're probably looking at
Mm-hmm.
specific things that are not necessarily core to being a plant per se, or
Mm-hmm.
or whatnot. And so what are those kinds of, like, how much of that is genome is it that you're looking at? Like, is it a very tiny piece where you're trying to kind of map the higher plants their function back and forth to the algae?
I would say it's a pretty large chunk. I don't have exact numbers off the top of my head, but um, yeah, I would say there's a pretty large chunk of algae kind of specific genes. Although yeah, you can infer a lot from um, kind of those gene annotations or those gene um, functions that are similar to plants.
Now this could this technique, do you think you're demonstrating it on algae, but do you think that it would translate to other species as well? Uh, could this go into the animal kingdom, for instance?
I mean, yeah, hopefully. I would say that. So I talked about how more and more eukaryotes they've identified gene clustering, right? And so it's been pretty common in certain fungi, specifically, and plants, and they've even identified it in a few instances in animals. Um, so I'm really interested in if I were to take this method and apply it more broadly. And hopefully in the future I am able to apply it more broadly. I believe there have been some studies that have um, applied this technique in 2 dimension to a much more broad set of fungi and animals and other species, and they have found several gene clusters, and that's available online to look through. I think it's called evolclust. Is the program that has kind of pre-computed a bunch of these gene clusters. I think they're still kind of there's so many that they're not all analyzed, but it's available from a database where you can look at that.
Do you have students working on this project?
Yeah, so I typically have undergraduate students. A couple undergraduate students will rotate through my lab each semester as part of research credit or hired off the grant. Um, so I've got usually 2 to 3 undergraduate students. I have some graduate students that also rotate through my lab, and I've got one that's joining me kind of full time in the fall semester. And yeah, so they're really helpful when it comes to right now I've got them working on kind of collecting the input data, um, formatting input data so that it's ready to be analyzed by Python programming.
Yeah, and that can be very time intensive, I know.
It is. So it's a lot of like organization and kind of getting everyone on the same page so they're not doing something that's already been done. Um, but yeah, and then after I run the program, get a bunch of clusters, I'll have the students work on data analysis and kind of looking into the clusters specifically in more detail, and that'll obviously be a lot of data to go through as well.
Now, this research you mentioned originated as 2D in a national lab. So I was just curious if you could explain to the audience what it's like at a national lab, just out of that's sort of my curiosity, but I imagine that the audience would be interested in as well. What's it like at a national lab?
Yeah, so I mean, I think it's kind of people describe it as a mixture between academia and industry. Um, so you do have individual PIs, principal investigators, that lead their own labs, but you also tend to be involved in these much larger multidisciplinary projects that those papers that might have 50 people on the author list, um, which I hear is more similar to industry. Um, but yeah, they are able to tackle kind of broader topics, more long-term experiments, because they do have, government supported funding for an extended period of time. So I tended to be on. Well, I did two different, very different things in the different national labs. So one of them, I was on this huge project, working with many different departments, different institutes, and but I also, found some time to be working on kind of the two-dimensional clustering, which was something kind of like a side project I was working on by myself. So I was able to do kind of both, work on a broad project, work on a really focused project, a lot of different projects going on. But yeah, I thought it was a really great place to do postdocs. The research is great. You really get to focus in on your research.
Thank you for that. That was something I find quite interesting. Now, as you know, this podcast is also about, of course, how the money is being spent. So can you just give like a brief overview of what you already mentioned that you're spending some on students and stuff. Is that
Mhm.
where the majority of the money goes? Do you have to buy computation time? These kinds of things. Where does the money go?
Yeah, so I would say the majority of the money definitely goes towards student stipends, um, specifically the graduate student, and then um, two undergraduate students I'm able to hire off this grant. So that's the majority in terms of kind of materials and supplies. Uh, Laptops have been where the majority of that money has gone to, for to enable the students to be able to, do the computational work. And what else? Supplies. We're going to be doing some sequencing of, like I said, some species that I believe was important to kind of fill in the phylogenetic tree. So we're going to be growing some of that species, sending it for high C sequencing, and then as well as there's some funding to attend conferences. There's funding to publish papers off of this grant. So
Yeah, and
yeah, I
publishing
think
papers can be quite expensive in
right.
certain circumstances. So yeah. Um, Now, uh, do you, because you are computational, how computationally intensive is this stuff? Like, do you sometimes have to dump it onto like a bigger machine, maybe that's hosted by the university or something, or do you mostly can it just be done on the laptop?
Yeah, so for the most part, it can be done on the laptop, which is why a lot of funding goes towards getting laptops that are capable of running these programs. So for the most part, most things are done on the laptop. There are some instances where probably when I the um, run the search for all the clusters, where I would need to use the university housed larger computational computers.
And um, a question that I always like to leave off on at the end is because of great interest to me is how did you get into science? So like if you could rewind back to, I don't
Mhm.
know, say middle school, maybe before or after, doesn't matter, but if you could rewind back to a moment and sort of identify what got you on this path, what was that?
Yeah, so I would say I was really into science fair projects when I was in middle school, high school. I mean, it was one of those things that our school made us do, um, but I was, actually really into them. And then by the time I was in high school, the school no longer made us do the science fair projects, but I was, voluntarily said I was going to sign up for an independent research class in high school to work on this science fair project. And this is the one that got me into algae in the first place. Um, I was looking at so I'm from Michigan, so at the time there was a lot of like invasive zebra mussels in the Great Lakes, and they ate a lot of the algae, and but because they were invasive, it was a problem. Um, so my experiment in the ninth grade was to look at, oh, can mussels kind of be a natural control to things like algal blooms, right? So I, kind of set up these tanks with mussels and fed them algae and would like count the numbers and try to keep them alive over the weekend. And 'cause I was doing all the work at in my like high school science classroom. So that was really my first um, kind of look into algae. I think I also did another experiment in high school with bioluminescent dinoflagellate phytoplankton, and it just kind of went from there. So it was something that was easy to work with. You didn't need any kind of special, super special equipment or clearances. So it was easy to work with, easy to obtain for a high school student. And then yeah, I just started getting really interested in algae and wanted to know kind of more how it worked. And then I started a PhD program where I went into an algae lab where we did more functional genomics, and that's where the bioinformatics started. So yeah, so I came in more on a biology side, and then kind of taught myself and took some classes and learned the more computational aspects, and then went on to do a couple postdocs where I have kind of transitioned more and more into the computational aspects.
All right, well, that was quite interesting. Thank you very much. And thank you for joining me.
Yeah, thank you for having me.